is basically an environmental microbiologist and biotechnologist. He did his master's in environmental studies from SES, School of Environmental Studies, QSAT, and he was awarded PhD in environmental biotechnology from QSAT from the same school itself. After that, he did his postdoc at National Institute of Advanced Industrial Science and Technology, Japan. And he has also worked as assistant professor at the National Center for Aquatic Animal Health, QSAT, for the MTech Marine Biotechnology course. After that, he joined as scientist at National Institute of Oceanography. Right now, he serves as the principal scientist at NIO. His research interest mainly focuses on the microbiology and biogeochemistry of tropical estuaries, mudbanks, open ocean, and coral reef. He's especially interested in studying host microbe interactions. He's in charge of the microbial reference facility maintained at NIO as part of the seawater quality monitoring program of MOES. He's a subject expert in marine microbiology for India, contributing to the second cycle of UN regular process for global reporting assessment of the state of the marine environment. He is the Indian project leader of India UK Collaborative Project Revival, as well as the India UK Japan Project podcast. He is an excellent researcher with more than 50 international research publications to his credit and has received several research grants and awards, including the First Track Young Scientist Award instituted by DST Government of India. Sir, we are privileged to have you as our speaker today. Hearty welcome to the Marine Biology webinar series. Over to you, sir, please.
Thank you, Swapna, for the nice introduction. And good morning, all of you. I thank Professor Bijoyanandan sir and Swapna and all other organizers for giving me this opportunity at this to meet you all at this platform. So let me share my. Yes. I think it's visible to all.
Yes, sir, it's coming.
Yeah, thank you.
Yes.
Yeah.
Good. Yes. Yeah, nice.
Yes. Now it is visible to all, and once again, good morning to all of you, and thank you very much for giving me this opportunity to meet you all in this platform. All of us, might have experienced water-associated disease at least once in our lifetime. So all the time, what we do is we attempt to correlate the same with food we ate in the previous day or water we drink. So we are all aware of this water-associated diseases, and my talk today is on the microorganisms or the marine microbial pollution in the coastal waters and its relation with the public health. So if you look at the global statistics of water-associated diseases, more than 3.4 million people die every year due to water-associated diseases, mostly from developed countries. And according to WHO, 1.8 million people die every year due to diarrhea, and 12 million are affected by typhoid. So these problems may be aggravated in future due to the lack of access to safe drinking water. Currently, there are nearly 780 million people who lack access to safe water sources. So India also is one of the country prominent with water associated diseases, and 1 out of 5 deaths, that is child death, are actually due to the water associated diarrhea. Apart from claiming lives, this water associated diseases also reduces the man working days, which can affect the economic growth of our country. So as per the Niti Aayog report, by 2030, nearly 40% of Indian population will be deprived of access to safe drinking water. So, to address these issues, UN has proposed and we adopted the Sustainable Development Goals, and there are several Sustainable Development Goals, and among which the Goal 3, which deals with the health, and 6 for water quality, and 13 climate, and 14 is for marine life. These are the SDGs which in which we are directly involved, or we are directly involved. The SDG 6 actually is providing access, deals with providing access to safe drinking water, sanitation, and hygiene. It also aims at integrated water management systems, and thus avoid pollution and protect water-related ecosystems. So international cooperation and capacity building have also been initiated to achieve these goals. So this slide, it is known to everyone, I will avoid it. And see, among the water-associated diseases, cholera is a major concern because it has already initiated the 7th pandemic and is still not under control. In many countries, there are nearly 1.3 to 4 million cases of cholera reported every year, and WHO put several efforts to reduce the death due to cholera by 90% and eliminate the disease transmission by 2030. I said only they eliminate the transmission because the cholera are present in the environment, so it cannot be removed completely. So these steps involved, the steps proposed by WHO for this includes identifying the reservoirs of Vibrio disease causing a pathogenic Vibrio, and then promoting the WASH protocols, and then again vaccines. So you may wonder why we discuss about, or how this is connected with the coastal ecosystem, coastal waters. So if you look at the history of public health, or the cholera incidents, in most of the cases, the first incidents of cholera are reported from coastal regions, and then it spread to the inland. That is the history, that is the trend which has been observed in many of the cases, and this could be due to various reasons. More than 50% of the global population and 15% of Indian population lives in the 100-kilometer periphery of the coastal regions, and this population density is going to increase with too many, including the Sagarmala project, too many developmental activities which are coming in the coming years to the coastal regions, and here is the availability of safe drinking water is also less in the coastal regions, and these regions are vulnerable to the food and surges, flood, and then the seawater surges during the monsoon, and the hygiene practices are compromised many times. So the marine environment can also function as a platform for the exchange of genes between autochthonous and allochthonous organism. I'm sorry, I will take a 1 second break. Oh, actually, my land phone was ringing, so I'm sorry for the disturbance. So this marine environment which receives effluents or the pathogens from several sources, and this waters can serve as a platform for the exchange of genes between the allochthonous and autochthonous organisms, and it is postulated in some studies that the calcium and this ionic concentration, the calcium and magnesium and the temperature which is present in the coastal waters may promote the gene exchange between these organisms. That is actually the favorable condition for the exchange of genes. And to exaggerate, I will say that the virulence genes and the antibiotic resistant genes can be exchanged. And so we will be, so that is why we need to discuss the coastal pollution in the region, and we will be discussing these aspects today. And again, for the examples, I can say the Vibrio infections or the gastrointestinal wound infections are increasing these days. For example, is 41% increase, I think I remember. So over 10 years have been reported from the US alone on this water associated diseases. Same is the case if I can show you this is the acute diarrheal disease which have been reported from the regions around Vembanad Lake. We can see that in the last, this is a 10-year record, which is collected from the health department, and we can see that the number of water-associated acute diarrheal diseases are increasing in the last 5 years. And interestingly, these infections are now being reported from regions from where it was eliminated long back by providing safe drinking water. That includes the Israel, it is reported, and some parts of US and Europe it is being imported, it is being reported again. So this is a condition. There are at least 12 species of Vibrios which can cause diseases, and the big fours are the Vibrio cholerae, Vulnificus, Parahemolyticus, and Alginolyticus. And these infections are, mainly through consuming the seafood or getting in contact with the water, coastal waters. So, and there are nearly 170 million endemic and respiratory illnesses which have been attributed to swimming in coastal waters or consuming contaminated shellfish, contaminated fish. And see, I will be using, to continue with the discussions, I will be taking some examples from 3 of our research projects which have been implemented in NIO. One is a seawater quality monitoring program, as part of which we are maintaining a microbial reference facility, marine microbial reference facility, which is funded by Ministry of Earth Science, and it has been running since 2003. And after that, we have I will be taking examples from our recent projects of Revival. It's an India-UK. It's an India-UK collaborative project under the water quality research programs. And the podcast is another India, Japan, UK. And these two are the examples of the international efforts we have been taken in the recent past to address the diseases. And when we discuss about the cholera, we have been hearing more about the climate change and how the climate change promote the water-associated diseases or the cholera diseases. Actually, the discussions on climate and environment relations on cholera is more than a century old. And the proponents of this concept are actually called as the localists, who argue the role of environment for environment and disease. They say that the pathogens are present, the cholera are present in the environment from where it can be causing diseases under favorable conditions. And while the other, the old theory was a contagious argument, who says that the man and the sanitation conditions are important in the transmission of cholera. But in the recent times, there are several examples in which the Vibrio cholerae has been isolated from the coastal waters, and environmental influence on the proliferation of this cholera Vibrios are much detected in the recent times. The environmental distribution and its proliferation or the transmission can be influenced by changes in the climate forcing and environmental conditions. So this can vary over, the transmission of the disease incidence can vary over different geographical time scale. For example, if you take the cases of Vibrio infections in the in different seas, different Punjab or Madras and the West, the Bangladesh mostly, we can see that it is influenced or it varies with the monsoon or the rainy seasons, and the temporary seasonal variations have been observed in the in the in Bangladesh on the Vibrio infections. And the climate variables actually can influence, or the forcing or the climate forcing can actually influence the sources and proliferation of pathogenic microorganisms in the coastal ecosystems. For example, due to the extreme weather conditions like flood or during the landslide, such incidents, the water body mixes up. It mixes up with the fresh water and the sewage waste, everything mixed up. And this is one of the examples which we experienced during the once in a century flood, which hit at Kerala in 2018. You could see that these are some of the images we took during this, and we can see that even this water hyacinth could be seen in the well water samples, and the turbidity increases in many cases. These are all due to the mixing of this. So this is promoted by so that the sources of pathogens into the environment are increased by these extreme conditions. And similarly, the proliferations of this pathogen also are influenced with the temperature or the climate change variables. So best example I can say is the seasonal variation and influence of flood on coastal ecosystem. That is the increased distribution of fecal pathogens in the Vembanad Lake during monsoon season. This is the real-time PCR result, which we analyze the E. coli using the real-time PCR. E. coli in Vembanad Lake using real-time PCR in samples taken at 20 days interval. We can see that the E. coli count, the fecal contamination is much higher in the Vembanad Lake during the monsoon season. This is actually during the flood season, so that it is much more higher. And I didn't mean that it is reduced to no detectable level after that. No, nothing like that. The contamination is still there during the months, during the other season also. But the fecal contamination is much higher during the monsoon. So how does it happen is a question. Because it could be during the monsoon season. It could be due to the flood or due to the terrestrial flow that fecal contaminants are reaching the estuary, and during other seasons also it is present. That means there are some leaking points are there in our sewage treatment systems in our city or nearby areas. And interestingly, if you look at the ADD incidents, monthly acute diarrheal disease in the regions around the Vembanad Lake, you can see that the number increases during the monsoon. So these two are showing some relation. So the environmental distribution of the fecal contaminants, or that environmental microbial pollution in the environment, and microbial diseases, water-associated diseases are related. At least in our studies, we can see with the Vembanad Lake and its regions. And more alarmingly, I will say that we isolated nearly 400 E. coli from the health indicator bacteria and studied the antibiotic resistance, and we can see that most of them are, more than 50% of them have high antibiotic resistant index, and sometimes it shows complete resistance against many antibiotics which are tested. And then the increased salinity salinity is one of the factor which has been considered as the climate variable, or the factor. Salinity and temperature are the factors which are influencing the distribution of Vibrio cholerae, and studies showed some correlations. And our own studies shows that it is not an individual parameter, but it is a combination of different parameters which influences the distribution of Vibrio in the environment. But most of the studies, they try to relate the salinity variations and temperature variations with cholera infections. And if you take the example of Vembanad Lake, we could see that, or the studies from NIO, the physical oceanography studies, showed that the estuary flushes out approximately 43 times a year, and that is every week during monsoon season, and during the pre-monsoon and post-monsoon season, the flushing is reduced. That means the salinity influence varies spatially and temporally, and this can have some influence on the distribution of pathogens. And the increased salinity which we observed during the pre-monsoon could definitely kill the fecal contaminants, E. coli, but that is not the case with the Vibrio cholerae causing organisms. That our studies shows that, well, generally the studies shows that Vibrio can tolerate the salinity, and the gene copy number, you can see that it is optimum is the 10 to 15 pSa salinity is optimum for the Vibrio cholerae. And this study is very interesting because this one, we use the cholerae pathogen in this study, and this pathogen was isolated from the, you may remember that 2016 there was a cholera disease outbreak in Kerala, and from that the causative organism was used in this study. And another variable which can affect is the which has been shown to have some influences on the growth of Vibrio cholerae is the temperature. And several studies reported, and the studies, Vibrio cholerae has been called as a barometer of climate change by Becker Austin. And several studies, including him, showed that 10 to 30 degrees Celsius are suitable for the, the growth of Vibrio cholerae. And if you look at this data, you can see that the Vembanad, this is the in situ and remote sensing data of Vembanad Lake. The temperature is always, within the lake in the range of 26 to 36, and though the average temperature, but very rarely goes above 32, but it is always in the preferable range. And the laboratory experiments conducted by Matena et al. And colleagues, and they showed that 28 degrees Celsius is optimum temperature, but we take the, this may vary between the environment and the laboratory conditions. But we can take more interesting results of Takemura, who studied, he compiled the data from different studies, and he suggests that the temperature higher than 30 degree might be a little bit, might have an inhibitory effect on Vibrio cholerae. So this is the status. Anyway, the study indicates that higher water temperature under climate change would favor the proliferation would favor the growth of Vibrio cholerae, and so their migration to new geographic areas in the temperate waters, and which can lead to 10 in a higher reported case can be expected in the future if the temperature is increased. So anyway, this temperature and salinity have been shown to influence attachment rates of Vibrio cholerae to planktonic crustaceans such as copepods, also have been reported in the literature. So this Vibrio cholerae actually can be found in the now we come to the importance of Vibrio cholerae and its environmental factors. But now how this Vibrio cholerae or the Vibrios can be found in the pollutants the microbial pollutants can be found in the environment. I can say with the examples of Vibrio cholerae which can be found in association with they can be free swimming. First of all, they can be free swimming, or they can be found in association with phytoplanktons or the zooplankton. This schematic representation shows how this Vibrio cholerae is found in association with and its participation in the biogeochemical cycle in the marine ecosystems. The bacteria can be found interacting with phytoplanktons or zooplanktons or benthic organisms or fishes or the particulate organic matter or the dissolved organic matter. And benthic organisms, it seems the crustaceans or the bivalves are also all are the the preferred host of this organism, this embryo, and they're also found in association with birds and water hyacinth also have been found, and the environmental reservoirs, well, then they may support the host pathogen interaction with the host, providing a source of nutrients and protection. And then the transport mechanism, it can serve as the transport mechanism of these pathogens, allowing its dissemination of this bacteria over a long distance. Then it can be the vector of infection, allowing the transmission of pathogen to humans, or even this Vibrio cholerae is also known for the role in the biogeochemical cycle in the system. So now let's if we discuss more on the reservoirs, you can see some studies, I can say they said that the sediment is the reservoir of Vibrio cholerae, from where it is coming up to the water column during resuspension. That is seasonally, but our studies showed that, I will show you the image later, that such an association was found with sediment. Vibrio cholerae was found in very minimum times in the Vembanad Lake, but but we found it associated with benthic organisms also. And the benthic organisms can be pathogenic. This Vibrio cholerae associated with benthic organisms can be pathogenic to themselves also, because some of the best examples are the the Vibrios affecting the shrimp, causing shrimp diseases. And there are many pathogens associated. And interesting things I should say is the association of this Vibrio cholerae with this migratory birds and chironomoids larvae. So this, when these birds swim in this contaminated water, and then this Vibrio may be attached to the body of these birds, and when they move to the other regions, it can be transported. And such type of examples are there for this case. And again, the chironomids have been reported. That is some insects that lays egg in the dry surfaces in the water bodies, and which will be colonized by the Vibrio cholerae or the Vibrio. And when this eggs hatch out, and these insects larvae are taken away by the airlifted, and it can be taken up to the other regions, and along with this, the Vibrio cholerae also be transported, so that it can cause diseases in geographically distant locations. And similarly is the case of water hyacinth. It can move from the fresh water to the, if you consider the fresh water, and if you consider the Vembanad Lake, you can see that in Tannirmukam region or the Periyar River, where they join, and during the monsoon season, you'll see a lot of water plants are there, which can move to as the salinity increases, it decays. But the cholerae Vibrio pathogens may not decay. It can survive in the lake, and the conditions may support the proliferation of these pathogens in the lake. And See, much studies of have been done in the case of Vibrio cholerae or the Vibrio association with phytoplankton and zooplanktons. Mostly the zooplanktons have been studied, and phytoplanktons also are there. And phytoplankton, I should say, I should say an example that uh, the example I can say is the case of Anabina, in which the heterocyst is there, and in which the microaerophilic environment is required for the activation of nitrogenous enzyme within the heterocyst. That is provided by the heterotrophic bacteria specialized to live inside the heterocyst. Similarly, there are a lot of examples where the heterotrophic bacteria, cyanobacteria interactions are reported. In one of our previous studies, you can see that this is a photomicrograph of a Cyanocystis isolated from Vembanad Lake, and this you can see the field emission SEM image in which you can see the bacteria attached to the Cyanocystis. And the interesting point is we isolated bacteria at Cyanocystis enriched from the polluted and less polluted region, that is upstream and downstream of the Vembanad Lake. We could see that it varies. The microbial diversity associated with these two Cyanocystis or the cyanobacteria varies drastically, and our further experiment showed that these, the one which is isolated from the polluted upstream environment have a large number of metal-resistant bacteria, which may be supporting or which could, play a significant role in protecting or ensuring the survival of this one in the metal polluted environment. So that is one thing. In the case of Vibrio, relation with the phytoplankton and or zooplankton, this can be negative or positive. For example, in some cases, some diatoms such as Skeletonema have been reported to produce secondary metabolites which can kill the Vibrio. On the other hand, some other phytoplanktons like Anapina has been shown to, reported to support the persistence of Vibrio. This association with this Vibrio and phytoplankton can be mutualistic. So Vibrio living in the polysaccharide sheath might take nutrients from the polysaccharide. In turn, it releases the carbon dioxide, which can be utilized by the phytoplankton for photosynthesis. This is one of the hypotheses proposed by many, and this association can be correlated with the presence of HAP gene also, and the association of this Vibrio with the zooplankton also has been reported from several studies, and we also, in our Vembanad Lake studies, we also found that the Vibrio cholerae are found either as in free living most of the cases, and or in association with the phytoplankton and zooplankton. And this varies between the seasonal variation is there and the spatial variation. That is, we classified the region into saline water dominated and freshwater dominated. Saline water dominated means in the Barmouth region and up to somewhere near to the Tannirmukham Bund, the salinity may go up to 15 and above during pre-monsoon time. So that is saline water dominated, and the Tannirmukham Bund and the adjacent area, some stations are classified as the freshwater. And this between these regions there is a difference in the association, and this difference and spatial difference also, and the seasonal difference also is there in the attached the abundance or the presence of Vibrio cholerae attached with the phytoplankton. And what this difference is, it could not be the salinity difference or the temperature difference alone, but it can be the the phytoplankton diversity or the community structure changes spatially or seasonally, which could also influence the interaction of this Vibrio cholerae with the phytoplankton, because some phytoplankton some phytoplanktons may be promoting the growth, and some are not promoting the growth of Vibrio cholerae. And if you and this most of these cases, the studies also correlate this with the presence of presence of this Vibrio with phyto or zooplankton is depending on the laminariness. So their ability is to colonize on the chitin cell wall, chitinous cell wall of the organism. So those zooplanktons which have chitin can it will be attached with the Vibrio cholerae. Similar is the case with the laminarin, and we can see that in most of the cases we isolated nearly more than 100 isolates of Vibrio, 100 number of Vibrio cholerae from the coastal regions, and we found that in more than 80% of the isolates have a very high laminarinase and the chitinase enzyme. So that must be facilitating their facilitating their association with phytoplankton and zooplankton. That is why we found that the Vibrio cholerae are associated more with the phytoplankton and the zooplankton, the Bibland lake. Of course, they are found as free living also. So if you look at the plankton exposure, we could see that it varies spatio-temporally in the lake. So here is the importance of the relevance of the observations made by Islam and his group and others. And they say the association varies with different phytoplankton. That we also have found the spatio-temporal variations, and which would contribute to the association of or the distribution of Vibrio cholerae in the Vembanad Lake. And now we need to monitor the Vibrio cholerae. So now we know the Vibrio cholerae are present in our systems, and that can cause the diseases. And now we need to monitor the systems. So what we have done in the past, recently, there are several programs have been done. And one is recently you must be hearing more about the Blue Flag program, Blue Flag certification of this beaches in the region, which also includes monitoring the microbial pathogens or quality of the beaches. Actually, this certification attempts to assure pure water, clean cost, safety, and access for all, in which the clean cost also includes the monitoring of E. coli or the fecal contaminants in the water column. Now, before that itself, we have started our coastal monitoring program. Actually, the seawater quality monitoring program was started in long back, and it was around in 1998. A group of scientists led by Chandramohan sir and they realized the importance of monitoring or monitoring the pathogenic organisms, health indicator, or the pathogenic organisms, or the contaminant, microbial pollution in the coastal region. So they included this Vibrio, the indicator monitoring in 1998, and the seawater quality monitoring. It was initially COMAS program, which included this microbial pollution monitoring in 2003. And in this program, NIO also was a part. And actually, as part of this program, we initiated Marine Microbial Reference Facility, which was actually initiated to select the media, the tools for monitoring, and then educate or train the others institutes, project fellows and research scholars at that time to monitor. And several training programs have been conducted, and support the final analysis while a polyphasic taxonomy of this pathogenic organisms have been included in that program. And then it came, they started thinking of the fecal contaminants. Total coliform came to the picture, and indicator organism, because they selected several indicator organisms because there is no universal health indicator organism, bacteria, but a number of bacteria, each with certain characteristics can be used. And, that should be able to, indicate its elements that can effectively monitor to approximate the risk of contaminated environment, while the indicators themselves should not cause necessarily cause any disease to the human. That was the criteria. So we know that a good indicator microorganisms should have certain qualities like easy to test in the lab or present in a greater number than the pathogens, because sometimes the many pathogens, including the viruses, their load may be very less in the water column, which the load that is enough to cause the disease much lesser than in detectable limits, so that we need to have some health indicator organisms that should be safer for the persons to work. So we came to the fecal coliforms, and E. coli are one of the most widely used fecal contaminant indicator organisms, but there is two school of thoughts. One says that questions whether E. coli is a good indicator because they can grow in our waters, that is tropical waters. They can climate conditions, they can grow higher temperature, so that there is two school of thoughts, and new anaerobic bacteria have been coming up as another promising indicator organisms. And when we started the microbial reference facility, these thoughts were in the initial stages, this hypothesis, and then they used coliform and Vibrio measurements were there, they promoted the use of high chrome media, that is chromogenic media. Based on the color formation, we could detect the bacteria, and at that time they also realized that there are different organisms. Say, for example, you can see that Clivera and E. coli can show the same color in the E. coli specific chromogenic agar. So that what to do is we may need to differentiate, so that in between the isolates, because there are many institutes involved in its monitoring, they used to send the isolate to NIO, and we have installed a facility called the microbial identification system, in which the gas chromatography was used to identify the fatty acid profile of microorganisms, using which we can differentiate very clearly the Clivera or the E. coli species based on the fatty acid profile. Now, these days, we are not much using this technique because the sequencing technique has much advanced, and everyone can use that technique very profusely. And but the chromogenic media non-specific problems are still there, so that in the past few years there were other techniques. For example, it's a fluorescent in situ hybridization technique. This is some of the optimizations we did, and identified the probes which can be used specifically. We can measure this fluvialis, cholerae, mimicus, and specifically using the fluorescent in situ hybridization, we can see and count the cells, so that the errors can be avoided to a large extent. And again, this was standardized and transferred to other labs for practicing during our training programs. Again, the techniques has advanced much in the last few years, and the real-time PCR technique have been introduced, and that is what we are using in our recent studies. We also don't use the chromogenic media right now, but we use of course we use it for isolating the bacteria, but for enumeration of the counting, we use the real-time PCR techniques. And now this marine microbial facility was created in 2003, and up to now we have more than 1,500 microorganisms isolated from different ecosystems. And when we started isolating, of course, this was the first one for health indicator bacteria from coastal waters. And later we thought, of course, this is a marine, and we elaborated the microbial reference facility to marine microbial reference facility, in which we have isolates from all the ocean, because we all know it is very difficult to get the sample from marine environment, open ocean cruise programs. We have to organize and get the sample, and we cannot repeat the samples very easily, sampling very easily. So we elaborated our cultural collection so that we are stocking the microorganisms received from the marine environment. But nearly 50% of the library is health indicator bacteria. That includes several pathogens, including the Klebsiella, Aeromonas or Salmonella, Staphylococcus, and Vibrios or Pseudomonas are there in the library. And this is available. And now, all this SWQ program also was, it's a broad program in which the total pollution was monitored. Okay, but later we realized that it is not the total pollution, chemical pollution, but microbial pollution itself have a great value, because we are going to face a real problem in future with the water associated diseases. So we need tools. For example, WHO also says we need tools to monitor, the contaminated areas. When we use real-time PCR or fish technique, these are all labor-intensive or technique-intensive programs which can collect samples from specific locations, one or two. Okay, and these samples need to be analyzed 1 day or 2 days needed, and we will get a very limited information. But now, what we need is, ensembled picture of a area where we can say that this area is contaminated, this district is this water body, this region is contaminated, and for which we need more sophisticated tools. And if we can do two things, that will be wonders, that can create a lot of change. One thing is whether we can use the remote sensing, and the second thing is whether we can equip the public to do the monitoring. These two can only solve our problems. So with this objective, we created two programs. In both of these programs, the first one we took it as a trial program. Vembanad Lake was monitored, and in which we use a combination of tools and experts from monitoring the water quality using in situ, and then we can use the drones for monitoring, or we can use the remote sensing for monitoring, and then we make the algorithms. So I will share some of this with you before stopping. And globally, this is a strategy that it moved from in situ measurements to remote sensing, and even the mobile GPS mobile based tracking of pathogen. That is recently in our COVID testing also they have used that a patient, infected patient, movement of the patient can be monitored. This was first used for the cholera monitoring in Bangladesh, because in the history of cholera, it was transported from India, or the source of origin, to globally through the transport, tourists, or the movement of public people. So this can be tracked with the mobile phones, or the distribution of pathogen can be monitored with the remote sensing tools. These are the initiatives which have been done in the recent past, that is in the last 5 or 6 years. This has been into the limelight. And when we take into the remote sensing, this is this image is taken using the remote sensing satellite. It's a private satellite has taken this image. You can see the Vembanad Lake. You can see even the Tannirmukham Bund is visible through this, and you can see these are all the water hyacinth, or the water plants, which is floating water plants, can be observed in this satellite. So that means the resolution is much higher with the remote sensing. But of course, the problem is more complex, because you can see the water plants or the bridge, or up to that resolution, you cannot see a microorganism. with the remote sensing satellite image. So that what we need to do is we need to make some proxies for for monitoring this pathogen. That's one of the proxy is the water plants, and several studies have shown that the Vibrio cholerae, for example, can be attached to the uh, to the the roots of these plants, and it can move. And similarly, it's the case with the phytoplankton, and the phytoplankton species or the diversity, the change with the phytoplankton type also have been shown, so that we can say these type of phytoplanktons uh, are the preferred host of Vibrio cholerae, and its abundance or its bloom or its presence can indicate some sort of cholerae present. So that can be correlated with the chlorophyll, and then it can be measured with the remote sensors. So that is the crisp of the approach which can be followed. But this problem is very complicated because you need the support of microbiologists, and then the physical oceanographers or the remote sensing expert, the modeling experts. These are all needs to be, or the marine biologists and the water chemists. Everyone have to come to the same platform to make this practically feasible. So that is a challenge, and that is the importance of multidisciplinary research, I will say. But you know, the results are much promising because the problem is global, and it is not limited to Vembanad Lake. No, we are we can, the same algorithms or same tools which we optimize can be used for globally. So that seeing this interest, we have built a global network of, of scientists who can interact. And then you may ask how we correlate with, and we did some preliminary experiments. See, this is this is in situ measurement of chlorophyll, and this is remote sensing measurement of chlorophyll. And now what we did is we measured the phytoplankton associated Vibrio cholerae, and then we could make some correlation between the chlorophyll and environmental Vibrio cholerae risk, probability of environmental cholera risk have been made, and the relation is linear in one region, freshwater dominated, but in the case of brackish water dominated regions, it's not linear. And this difference in the relation, this relation can vary between regions because the phytoplankton community, the chlorophyll composition, everything varies, and that has to be optimized with large scale measurement. Anyway, this is, you see, this is the chlorophyll image, and this is the risk map of cholera, which we could prepare. This is a attempt, and a lot more have to be done to optimize or make this foolproof. Efforts are going on. So, but in near future, we can have a risk map which can be prepared every 2 weeks or every month, saying that this region is contaminated, so that we can avoid coming into contact with this region, or we can take some more precautions to avoid the infection. So that is how we can avoid the spread of or the transmission of Vibrio cholerae, and that is what WHO also is aiming to implement, and SDG also is aiming to implement by 2030. So, but as I said, remote sensing alone will not solve our problem. Okay. Public participation, especially the any diarrheal disease or water associated diseases, most affected is the women and children. And if you see, I said in the first slide, 1 out of 5 death in child death in India is due to water associated diseases. So we need to educate the public. How we educate, we have been doing the awareness program for a long time, but awareness program and citizen science is different. Citizen science is something like we need the public or the citizen is involving in the monitoring, and they have some tools. So what is the most important in this is you need to provide, you cannot give a real-time PCR machine or the PCR machine to the public and ask them to monitor every day. No, that's not possible. So they need some tools which can be used regularly. Okay, and that can be, if it is a pocket-sized tool, that is wonderful. And that is the area where we did some combinations. like, 90% of the public believe the water they drink or water they take bath are safe because it looks clear by their observation. So now we need to correlate the clarity with the contamination. That is the objective. So in that objective, we did a nice combination. This was made by the Plymouth Marine Laboratory, and in discussion with us. And this is a 3D printed pocket sized device, and in which two equipments are merged. This is the Fourier scale, and this is the Secchi disk. So these two permit us, this one will permit us to measure the clarity or the transparency of the water column, and whether it is turbid or no, if it is turbid, the clarity or the Secchi disk depth will decrease. And this is a Fourier scale in which the water quality water column can be classified into different units, that is up to 22 units based on the color. This brown means it is the resuspended suspended particles, and this is phytoplankton bloom, and this is clear water. And these two tools are merged as a 3D printed pocket size. And now we train the students, nearly more than 100 students from different colleges, and the stakeholders when those who are living near the Vembanad Lake or daily commuting in the Vembanad Lake. Students are also commuting in the Vembanad Lake. And NGOs were trained, and they were given with nearly 100 such it is, so that they do the measurements. Now, and we also created or made a mobile application, which is Tarbaqua, in which these data can be transferred using this application. You can transfer the data to the server placed in our CMFRI, so that the online transmission, so that when we use the smartphone, we know the latitude and longitudinal position of the area, and then the data can be collected. Even the image can be taken and transmitted to the server, so that the data accumulate. And now I tell the beauty of this data. You can see that the yellow dots are the measurements which made by the student or the citizen scientist. And if you remember there, if I say that during the whole revival project, program 3-year, we had 13 stations into 20 days interval samples, nearly 250 or less than 500 samples altogether we collected. But now the citizens made nearly 1,000, more than 1,000. Now it is 1,200 or plus readings have been made by citizens, and it is all around the Vembanad Lake. And these are some exceptions, and this happened because this is from the land. No, it is not like that. It's happened because they took the reading from here, and their mobile was at home. So they transferred the data from the mobile from at home, so that you will see this reading. So then these problems are rectified, and now this has been taken from the Vembanad Lake. So this is the beauty. So 2,200 or 300 or 400 reading to 1,500, and this is over a 6 months or 7-month period. You can imagine how much the quantity of data which we get in over a period of 1 year or 10 years about the Vembanad Lake, and then our readings will be perfect. So that is how a day will come that the public and the citizens can go and monitor the Vembanad Lake, and the mobile application will tell whether they can take bath in the system or they can use this water. And this can be transmitted to anywhere, any water quality systems. And this is a map which we have been made based on. So if you are interested to join with this program, definitely we welcome you to join. And you can contact myself or Dr. Nandini Menon or Dr. Grinson George for getting further details on this citizen science monitoring. And now we have examples from here, and now this is a global problem. So we made a onboard platform. Okay, that's an open network platform of we made this platform for those who are doing research in Vibrio cholerae, environmental cholerae, and doing research in the field. They can interact, exchange data, and they can work together, or they can even have more projects together. So for gathering these experts and researchers, we made the platform in which our Madam Rita Colwell is a member, Biswat, all the those who are working in this field have become the board members, Biswat or Rita Colwell or Thompson or Craig Baker Austin. These are all the people who work in this Vibrio cholerae, and we made a group, and which is led by Milton Campbell from Brazil and Dr. Shubha Satyendranath, and she is also an alumni of our marine science, and this is funded by Global Challenge GCR of UK. And who can join this program? You can also join if interested. Join this program. If you are interested to join this program, definitely please shoot an email to onward.gcrf@gmail.com, and these are the people we are expecting to join with this program. So let me before concluding, if you want to get more information on any of the topics which I discussed today, and the talk is based on these publication, our own publications, which you can get it from our Google page or ResearchGate, or these are many of them are freely available, open access. And so I thank you all. Before thanking, I should say this is a tribute to our mentor who made this, Professor Trevor Platt, because when I went to meet Trevor Platt in 2016 or 17 to discuss, my aim was to do the monitoring using the real-time PCR and some modeling part, but it is a discussion with him. The team elaborated, and now we have a lot of people remote sensing, modeling, and it becomes a global program. So this is a tribute to Trevor Platt. So thank you very much. I hope I finished in 1 hour. So thank you, Swapna, and all for listening for the talk. So
Thank you so much. The floor is now open for discussion. If you have any doubts or queries, you can ask, or you can just type it in the chat box.
yeah, please. Well, if there is no question to ask, and if you get some doubts in letter, you can email to me also. No problem.
Yeah. Can I ask Dr. Swapna? Can I?
Yes, sir. Sure, sir.
Yes, sure. Dr. Anas, it
Yes,
was very composed and very systematic presentation, largely focusing on your publications and the research output, how microbial ecology is very important in ecological management and ecosystem-based management of large coastal ecosystems, especially for coastal ecosystems. I don't have much doubts on that, but I was just looking, other than Vibrio, what are your focusing areas? Because you largely focused your presentation on Vibrio, I think so. Am I right? Yeah.
Yes, sir.
Yeah.
Yes, sir.
So, I mean, because I think excellent relationship you have come across with phytoplankton, zooplankton, even benthic organisms, where there are associations and where the phytoplankton exudates, especially, are getting nourishment as well as they're affected also to a large extent. So other than Vibrio, what could be in that case, there could be a plethora of other microbes or microflora which will be influencing or which will be contaminating. Can you throw some light on that?
Yeah. Yes, sir. Yes, sir. Why I selected Vibrio is because in this talk is because it's a good example. When we use the examples, it is easy to convey the messages. Especially, sir told me that most of them are students. So I thought, it should not be a complicated. So I thought let's select Vibrio cholerae. And this, if the pathogen is, this is one of our research program in which we are focusing on Vibrio cholerae and Vibrio species. So it can be Vibrio vulnificus, parahemolyticus, all are there. And basically, our lab is working, as Swapna told in the introduction, we are working on the host microbe interaction, how the microorganisms interact with with their host, and what are the factors which are dictating these interactions in specialized ecosystems. So we work in lake, Vembanad Lake, oxygen minimum zones, mud banks, and the coral reef ecosystem. So these are the different ecosystem which have different nutrient profile and physical conditions, and human interferences are different. So we selected, of course, the deep sea is also there. So we get a comparison on environmental parameters. So we use different tools to study the microbial interaction. Like next generation sequencing, culture independent tools are largely used to understand the interaction of the diversity of microorganisms. But what we are promoting now, what I am proposing now is for the culture dependent studies, because independent studies will give some information who are all there. Something like the chemists speak about the periodic table. We can say there are some elements which have not even detected till date, identified till date. So, but when we want to translate the information, we need to work on organisms, attempting to isolate. So we are trying to isolate microorganisms using flow sorter techniques and a different combination of techniques. That is one of them. And we have been isolating, trying to isolate this Planktomyces type of organism from the oxygen minimum zones, and Lactobacillus or such type of different types of Vibrios also is there. Different types of organisms. Then in vitro experiments are being conducted to understand its interactions with the host and also in the biogeochemical cycle. Why we do this is the ultimate aim of the CSIR is to translate the ecological information to products and processes which can be useful to the industry. And that is where we need to have these organisms in hand. So this is the overall research our lab is doing currently.
Excellent. Nice. Thank you.
Thank you, sir.
Thank you.
So we have got a question in the chat box from Vignesh. How might calcium and magnesium ions promote gene exchange in bacteria?
Oh, that is actually, it's a hypothesis, because you can very easily compare it with our, if you are doing the molecular biology experiments, we prepare the competent cells. How we prepare the competent cells is the same thing in the, gene transfer, we do the same concept they have. Then when we have this, then the microbial pores will be opened in a bigger size, and through which the entry of these genes are possible. That is the hypothesis says about the role of this calcium and magnesium.
Okay, thank you. Sir, I have a doubt. You mentioned about the microbial library that have been maintained at NIO. Is it a metagenomic library or live cultures?
We maintain the culture. We maintain the culture. Isolates are there.
Okay. And what is the purpose of that, sir? Are you planning any future studies based on that culture, or is it just depositing whatever is the?
There are a lot of applications with respect to the microbial, marine ecosystem studies, and also we can see the progression of this, how this antibiotics. Say, for example, before going to that, this reference facility is available to everyone, so that now in one of my collaborations, I am sharing these isolates with IACT for they are looking for the products.
Okay, so we also have access to that
Yeah,
library.
definitely. We all have the access to the library, and also, this facility was open for MIS. Also is open for all the facilities available with RC. My team is open to all. Okay. Even we can see CSIR is have made all its facility available. That is CSIR Tech. I forgot the name. There is a website through which all the equipment facilities are available, open to all.
Oh, thank you, sir.
Thank you.
Any more queries? Vijay, sir, any questions?
It is fine. I think it was well presented. I think a lot of
It was
input.
really an informative session.
Thank you.
So, wind up the sir.
Yes, please.
Yes.
Yeah, so thank you so much for the interesting and informative session, sir. And also thank you so much for sharing your valuable time, knowledge and experience with us. Thank you, Sarana teacher. Thank you, Vijay sir, for coordinating all these webinar series. Thank you all the faculties, Dr. Priyaja, Dr. Srireka and Dr. Padmagumar and all other participants, the research scholars as well as students for joining us. Have a great day. Thank you.
Thank you all.
Thank you, Dr.
Thank you,
Anas.
sir. Thank you,
Very nice.
sir.
Great.
Thank you for giving this platform.
Thank you.
Thank you.